All right, you guys, welcome back. We're in the last section of Chapter 5, Beyond Mendel's Laws, and that is Section 3, looking at linkage. Now, we're going to talk a bit about linkage. We're not going to get into super detail. We're just going to kind of introduce the topic, all right? All right, so then let us get started. So linkage was discovered in pea plants. What they noticed is that in certain dihybrid crosses, they didn't notice, or sorry, I said notice a lot, didn't I? They didn't show a typical 9:3:3:1 ratio that would be expected when you have two heterozygous parents. What they noticed instead was an excess of the parental types and some recombinant types. What this told us is that it appeared that some genes travel together. Genes on the same chromosome are linked. So if they're on the same chromosome, they will oftentimes travel together. Now there are variations in that. There's the closer they are on the chromosome, the more linkage they have, the farther on, the less linked they are. And since they are linked, we do notice different patterns of inheritance than unlinked genes. Because when we're talking about previously with the dihybrid crosses, we're looking at two completely separate chromosomes where the genes are on them, and so you can have many different possibilities. But now we're looking at genes that can travel together because they're located on the same chromosome. The farther apart two genes are on a chromosome, the greater the probability that crossing over will occur between them. So remember, crossing over is when the homologous pair gets so close that they fuse and they can actually exchange pieces of information. So if we're looking at the pictures on the bottom, notice how close gene 1 and gene 2 are on the same chromosome. There's very, very little space between them, so it's very unlikely, it has a very low chance that crossing over will occur because there's not a whole lot of space between the two genes for synapsis when the tetrad forms and they fuse to occur in order to allow them to exchange information. Here, we have gene 1 and gene 2 on completely different chromosomes. So crossing over will not occur between them, therefore they are not linked. And the last one, they're still located on the same chromosome, but there's a much bigger difference, or much bigger distance, between them. As a result, crossing over has a much higher chance to occur between them. So they're not as linked. I wouldn't necessarily say they're not linked, but they are not as linked. So the chance of them being linked goes down significantly. So you mentioned, or I've mentioned parental types. Let's talk a little bit about parental versus recombinants. Parental types are progeny that have the same phenotype as one of the parents or of the other parent. Recombinants are what you get when crossing over occurs and you end up with progeny that do not have the phenotype of either parent. So if we look at this, we're saying the blue comes from one parent and the red comes from another. Now here they're showing crossing over. Down here, they're showing you the possible chromosomes that can be separated into the gamete cells. All blue, that is a parental type because it matches up here. All red is a parental type because it matches that one. But notice that we have some differences here. The parental type was big H little g, little h big G. Big H little g, little h big G. Big H big G, we don't see that in either parent. That is a recombinant. Little h little g, we don't see that in either of the parents. It is a recombinant. So parental types are the offspring that have the exact same combination as one of the parents. Recombinants are when they don't resemble either parent. So the combination of the alleles for those two genes are different than what the parents themselves have. This kind of gives you, again, another example. So here we have a wild type, which is the regular wings and the kind of grayish body. And here we have male fly who has a black body and vestigial wings. And this is showing you the possible chromosomes that this parent has. So one chromosome, big B, and then big B, G, sorry. So dominant, dominant, recessive, recessive. So this parent is heterozygous. This parent is homozygous dominant for both traits. Now, if we look down here, what we see is this parent can have a few different combinations. Now, since both of these have the same genes, it doesn't even matter if recombination occurs or if crossing over occurs, because they have the same information regardless. It's like, if I give you a $1 bill, you gave me a $1 bill, we both have $1 bills. We didn't change anything. But if I gave you a $1 bill and you gave me a $20 bill, we both gave each other a bill, but we got two different ones. So here we have a parental. This chromosome doesn't show any crossing over, neither does this one. But these two are the recombinant chromosomes. They do show the crossing over, which means we have different combinations. So we have dominant recessive, recessive dominant. And what we end up with when we cross them with the black vestigial fly, we end up with these possibilities. So 944 offspring will be black with vestigial wings. That's a parental type right there, black vestigial. 965 would be wild type. That's a parental type. 206 would be gray and vestigial. So they would have this one's body type and this one's wings. That's recombinant. And 185 would have black body and normal wings. So they would have this one's body color, but this one's wings, recombinant. All right. So we have these things called linkage maps. And what they are, they're based on observation to determine the likelihood of crossing over occurring between the two genes. And what they've noticed is that the distance between the genes is directly proportional to the likelihood of crossing over. Put that simply, the more space between them, the more likely crossing over can occur. The less space in between them, the less likely crossing over can occur. Can also build linkage maps by tracking these transmissions, by looking at the number of times we do see the recombinants between the genes. And we can use that to then figure out the percent recombination between them, meaning the percent chance that crossing over will occur. Now, if you notice here, between these two, it's saying there's a 9% chance. If we're looking at between these two genes, 9.5% chance. And if we look at these two genes on the outside, it's a 17% chance. They are further away. They have a higher percentage chance, which means that it's a more likely chance that recombination can occur through crossing over. Smaller distances, smaller percent chance that crossing over will occur. Bigger distances, bigger percent chance that crossing over will occur. This is a linkage map of one of the chromosomes and the traits on it relating to fruit flies. So these are looking at distances, not percentages, but they also show you the trait, the normal ones up here, and then the mutant varieties on the bottom. So it's just starting at the end, we'll consider that distance 0. Wings are distance 13, leg length, distance 31, 48.5, 54.5. Now, if we look at this, and we just kind of pick some of the different genes. If I'm looking for the gene for body color right here and the gene for eye color, that's not a big space. So because that has the smallest space between any of the genes, there is a very, very, very low percentage, or recombinant percentage, which means there's a very, very, very low chance of crossing over occurring in this small space. However, if I'm looking at the distance between the wing length right here and the wings being straight or curved, that's a much bigger distance. It's this entire distance right here. So that is about 75.5 minus 13, because the 75.5 is to the 0. So that would be 62.5. There's 62.5 map distance between them, which means that with that larger space, there's a much greater chance of crossing over occurring between here. So the likelihood of them being linked is low. The likelihood of recombination or crossing over occurring is high. As opposed to this one right here that we're looking at, which is 6 map units apart. Much smaller distance. So linkage in humans. Linkage data can be pooled from human pedigrees. So we're looking at human pedigrees from a lot of different people in order to figure out the transmission probabilities between genes that are located on the same chromosomes. And we've been able to use this to start making human linkage maps. Now we're not quite there yet. We're still working on this. We do have a lot of data, but we're still working on it. It's easier to create linkage maps for fruit flies because one, they have very, very quick generation, so it's easy for us to see them. You can go through multiple generations in a month, as opposed to humans, it takes a bit longer. We do look at something though called linkage disequilibrium, or LD. This occurs in the human genome, and what it does is it reveals non-randomly distributed sequences where recombination occurs infrequently. So what this means is that it's used to figure out if there is an association between alleles at 2 or more loci. Loci are simply gene locations. So we're looking to see if these two genes at these different locations, for whatever reason, are associated and that they always seem to travel together. Even if they're not considered linked, even if they're not close to each other, we still see them traveling together. It is related to the time of the mutation event, the genetic distance, and the population history. So it does help us to try and figure these out, why recombination occurs so infrequently between these, and helps us to better increase our map knowledge. Again, this is just kind of showing you like how this can look. This is just looking at the linkage and how we can see things being traced through. And this is showing the LD, how we see the shift in colors and the shift in variety. And then until we see more equilibrium, where it's more equal across the population. This one's just looking at how linkage can lead to these colors moving around. This is showing how it distributes from very different to very similar distribution. You don't have to know that much about it for my class. We just want to introduce the idea of it. All right, evolution in gene mapping. Yay for computers, right? So computer analysis of linkage data allows the computation of what we call an LOD score. So the LOD is a logarithm of the odds. So it's kind of looking at the chance of things occurring versus not occurring. In other words, how likely is it that these genes are linked versus not being linked? And it's on a logarithmic scale. So it's not like these are numbers 0.5 to 1, 1.5 not. Those aren't percentages or anything. But these computer analysis have actually helped us to better understand how linkage works and how to predict it. What they have noticed though is that an LOD score of 3 or higher signifies that there is a huge likelihood of linkage between the two different loci. So they're more likely to be linked and they're more likely to travel together. Haplotypes. What haplotypes are these tightly linked series of markers that appear to be inherited as a single unit. So what that means is that there are these, we can tag them, and if we were to put tags at these specific locations, they're pretty closely together and they will actually move together. Now, even if they're not super close together on the chromosome, they'll still stay together and they will move together. So they are linked and they will be passed on together as if they were a single unit. Now, we're still kind of learning about how these haplotypes are, what they do, how they function, and why they stay together. But we do believe that these markers may be protein coding alleles. So they're alleles that code for specific proteins, and that those are essential proteins. That might be why they travel together. Could be these things called variable restriction sites, also known as RFLPs. They could be these variable short repeated sequence, VNTRS, or single nucleotide polymorphisms. We've talked about those, what SNPs are, back in chapter 1. Again, you don't have to know this much information. You don't have to go into any more detail than this. We're just introducing it so that you have an basic introduction to these ideas, especially if you want to go on to study more in-depth genetics in college. All right, additional resources since we are at the end of our chapter. So here we have Science in Pajamas, so the ones I make. We do have one that talks about incomplete dominance and codominance, talks about what they are, how they're different, also gives some Punnett square examples. We have one on multiple alleles and blood types. So we're using the idea of multiple alleles, specifically with blood types, to talk about the different combinations, as well as what it means in terms of your blood type. And epigenetics. Sorry, that's supposed to say epistasis, and I will fix it. So I will get the epistasis video up there. I do apologize for that. So the epistasis video is not supposed to be the epigenetics, it's supposed to be epistasis. I will get that fixed for you. So by the time you guys actually see this for class, that will be fixed. But we will have a video on epistasis, not epigenetics. And then other videos. So we have Amoeba Sisters on incomplete dominance, codominance, polygenetics, and epistasis. It is epistasis, see in the video. Why did I put epigenetics? Too many epigen words. Amoeba Sisters, multiple blood types, ABO blood types, and Punnett squares. Steve Gorshik with lethal alleles, pleiotropy, and phenocopies. We have biotech review on mitochondrial DNA. We have Professor Dave Explains with gene linkage and linkage maps. Hey, look, it's the flies. And we have Bozeman Science, genetic recombination and gene mapping. All right, so there's all that. I will get that one Science in Pajama link fixed, so I apologize for that. But in the meantime, if you guys have questions, let me know in our Google Classroom or in email. And I'll post this link in the video description so that you can have access to those additional resources. But in the meantime, you guys just stay awesome, stay amazing, stay curious, and stay safe and healthy. Guys, you guys take care. Bye-bye.